Good morning student, myself Manoj Chotia from Raghunath Vidyalaya. Welcome to our new class. First of all, we want to discuss our syllabus because some part of our syllabus is deducted. We have total 42 lessons. Among them 14 lessons are removed by Board of Secondary Education. The lessons number I am telling you. First lesson, reproduction in angiospermic plant. Fifth lesson, plant water relation. 12th lesson, nitrogen metabolism and nitrogen cycle. 14th chapter, biotechnology, general introduction. 17th lesson, economic botany and human welfare. 19th lesson, sustainable agriculture. 21, integumentary system of human. 26, nervous system of human. 27, sensory organ of human. 30, movement and locomotion in human. 37, mutation. 38, human population. 41, domestication of animals. And 42, Jav techniques in medical science. So these lessons are removed. Now we have only 28 lessons. Among them, some topics are also removed in these chapters. So first of all, we discuss in 16 lesson. In lesson number 16, the achievement of plant tissue culture is removed in this chapter, and I am telling you that in each lesson some of the topics are removed. So that topic I will not teach you in our next classes. Okay, just like the lesson which we are studying here, plant tissue culture. In this chapter, culture media and application and achievement of plant tissue culture are removed. Only two topics in this chapter. First, culture media, and second, applications and achievement of plant tissue culture is removed. So and another lesson that we will study, full lesson, integumentary system of human, that is also removed from our syllabus. So you have no any difficulty. I just tell you all the lessons which removed in the syllabus and the topic which are removed in the syllabus, that we will not study in our next classes. Mainly you have to learn in this chapter, means chapter number 16, that we are studying here. So then I am telling you plant tissue in culture introduction, important terminology used in tissue culture, necessary resources for plant tissue culture, steps of micropropagation, types of culture, method of gene transfer in plants, transgenic plants, and important achievement in agriculture and medical science related to transgenic plants. So we are studying here that transfer of genes. Transfer of genes are of two types. First, indirect method. Indirect has three types. First, plasmid mediator gene transfer, then virus mediator gene transfer, and third, in planta technique that we will study, that was studied by us. Now today's our topic is direct gene transfer. Direct gene transfer has also two types. First, chemical transfer, and another physical transfer. Look at the board. So these are the two direct gene transfers method. First, chemical, and second, physical. In physical there are 5 types. First, gene, second, electroporation, third, liposome mediated gene transfer or lipofaction, fourth, microinjection, fifth, macroinjection. Now first of all, chemical method. In chemical method, first of all you see some examples of chemicals. PEG. Here 1 mark question is performed. What is the full form of PEG? Polyethylene glycol. polyethylene glycol, PVA, polyvinyl alcohol, polyvinyl alcohol, calcium phosphate, DEAE. What is the full form of DEAE? Liposome diethyl aminoethyl or dextrose protein. In the presence of these chemicals, cell and desired gene are put in a pot. Means, for example, here is the cell in which we want to inject desired gene, and this is desired gene, and this is the chemical. For example, this is PEG. In the presence of PEG, polyethylene glycol, the desired gene and the cell in which the gene should be transferred put in the presence of these chemicals, then automatically this desired gene is enters into the cell. So this is the direct gene transfer method or chemical method. Means, in the presence of these chemicals, polyethylene glycol, polyvinyl glycol or calcium phosphate or liposome diethyl aminoethyl or dextrose protein, in the presence of these chemicals, both the targeted cell and desired gene are puts. So this gene is transferred into a particular cell. So this is the chemical gene transfer method. Now physical method. First, gene gun. It is also named by so many names, just like shotgun method, particle gun, micro projectile. This method is discovered by Kelin. So you have to remember the name of the scientist, because here 1 mark question is performed. Who discovered gene gun method? Kelin first of all discovered gene gun method. Gene gun is just like a shotgun or a revolver. Whenever a gun is shooted to a particular person, same like that, whenever the desired gene are overlapped by gold or tungsten, then after by high pressure this tungsten or gold is entered into the target cell. So this is called micro projectile. First of all, in onion cells, DNA and RNA are transferred by these methods. You can see a diagram to understand this. First of all, we overlap the desired gene by tungsten or gold. Then after, due to high pressure, this is target gene. From high pressure, this desired gene is transferred into this cell. So this process is called gene gun or shotgun or particle gun or micro projectile, which is discovered by Kelin. Now next, electroporation. Whenever we want to transfer desired gene in a particular cell, then we give high voltage to this cell for a nanosecond. Then the cell can be poured in from many places. जैसे ही इस सेल को क्या करते हैं, हाई वोल्टेज प्रदान करते हैं, हाई वोल्टेज देते हैं, तो इस सेल में क्या होते हैं, अलग अलग प्लेस पे और पोर्स हो जाते हैं, और उन पोर्स के थ्रू फिर डिजायर्ड जीन जो है वो इसके अंदर ट्रांसफर हो जाता है. दिस इज कॉल्ड इलेक्ट्रोपोरेशन. थर्ड, लिपोफेक्शन और लिपोसोम मीडिएटेड जीन. जैसे जीन गन के अंतर्गत हमने क्या किया था, गोल्ड या टंगस्टन से ओवरलैप किया था किसको, डिजायर्ड जीन को. उसी प्रकार यहां क्या करते हैं, लिपोसोम मीडिएटेड जीन ट्रांसफर में वाटर और लिपिड के द्वारा क्या करते हैं, इस डिजायर्ड जीन को ओवरलैप कर लेते हैं और कोशिका के पास में इसको रख देते हैं। तो ये जो लिपिड है वो ये प्लाज्मा मेम्ब्रेन के लिपिड से टच हो जाता है और इसका जो डीएनए है वो इसके न्यूक्लियस में ट्रांसफर हो जाता है। सो दिस इज कॉल्ड लिपोफिक्शन व्हेनेवर द डिजायर्ड जीन इज ओवरलैप्ड बाय वाटर एंड लिपिड and then put them to the desired target cell. They are put together, then after the lipid is attached to the plasma membrane and the desired gene is transferred automatically into the cell. So this is called liposuction. Next, micro injection. जिस प्रकार से ह्यूमन बीइंग के एंटीबायोटिक के इंजेक्शन लगते हैं उसी प्रकार क्या करते हैं जो माइक्रो इंजेक्शन है उनके अंतर्गत इंजेक्शन में डिजायर्ड जीन को भर के और डायरेक्ट सेल के अंदर उसको इंजेक्ट कर दिया जाता है। एंड वेन एवर द लेंथ ऑफ डीएनए इज सो हाई एंड वी हैव टू नो दैट इन विच फॉर्म वी measure the length of DNA in kb. Whenever the length of DNA is high, then we took macro injection. मीन्स आप यूं समझ सकते हैं कि ह्यूमन बीइंग के जो इंजेक्शन लगता है वो छोटा होता है और एनिमल्स के जो इंजेक्शन लगता है वो बड़ा होता है। तो डीएनए की अमाउंट जो है वो ज्यादा होने पर बड़े आकार के इंजेक्शन में उसको भर के और डायरेक्ट सेल के अंदर इंजेक्ट कर दिया जाता है, ट्रांसफर कर दिया जाता है। अगर डीएनए की अमाउंट लेस है देन माइक्रो इंजेक्शन और डीएनए की अमाउंट हाई है देन मैक्रो इंजेक्शन। थैंक यू फॉर टुडे क्लास।